Great. Welcome to the first place winners call. Peter, I will turn it over to you to introduce the Novozymes team.
Sure. Thank you so much. My name is Peter Hallen. I'm heading a team of 6 full-time scientists, bioinformaticians, and protein engineers here in Novozymes. We are mainly involved in computational tools for engineering proteins and discovering new enzymes. So with me, I have Esben, as well as Morten and Jesper. But let's start with Esben.
Yes, I mostly work with 3D structures and designing variants of enzymes for new product candidates.
Yeah, and I'm Morten, originally a molecular biologist, but in the past 5-6 years, I've doing mostly bioinformatics, working on discovering novel wild-type enzymes.
Yeah, and hello, I'm not unknown. My name is Jesper, and I've been following the competition. I'm also working with bioinformatics and a lot of machine learning tools, applying that. And we have Roland as well, just joining.
Who is muted without camera, but there we have it.
Can you see me? Hi, guys.
Will you introduce yourself, Roland?
Yes, I'm also part of Peter's department. I just joined now, so sorry I missed the beginning. But I'm mostly doing protein design, data analysis, bioinformatics tasks, so a wide variety of things.
Thank you.
Yes, let's start my presentation. Okay. I am the first place in Novozyme enzyme stability prediction competition. I'm delighted to be speaking with you today. I'm going to talk to you about my solution. Let's start. This presentation will follow this agenda. First of all, I will give my background and the summary of my solution. Last year, I graduated from Kyoto University with a Bachelor of Science degree. I majored in particle physics, so I know little about biology or chemistry. But I'm interested in machine learning, and I worked as an intern studying machine learning, especially graph neural network. While studying graph neural network, I came to know proteins dataset. Proteins dataset is one of the most commonly used benchmark datasets for graph neural network. The task of proteins dataset is to predict whether a protein is an enzyme or not from the graph representation of its 3D structure. So I knew that graph neural network could be useful for protein data. I participated in this competition to see if graph neural network would be useful for this type of task. This is a summary of my solution. I use graph neural network, and I used ESM2 to generate node features which are input to graph neural networks. To implement graph neural network, I utilize a library named Deep Graph Library. The entire process of training takes 56 minutes. In this section, I will share the feature engineering I did. For target, as many participants did, I used the variation in melting temperature, DTM, as a target, instead of the melting temperature TM itself. Since only the ranking of the sum stability is relevant to the score, this change doesn't matter. And how to use the information contained in PDB files is one of the key points of this competition. PDB files contain the information on three-dimensional structure of protein. I converted the 3D structure information into a graph. A graph consists of nodes and edges. A node represents a residue. And if two residues are closer than a certain distance, 6 angstrom, the nodes are connected by an edge. This is a graph of wild type of test enzymes. Each node in a graph is able to have features. As node features, I use the embedding vector generated by ESM2 from the amino acid sequence, and also use encoded physical and chemical properties of each amino acid. In this section, I will share my model. I utilized graph neural networks on the protein graphs. Graph neural network is a type of neural network that handles graph data. Node features are sent to neighboring nodes through edge. This operation is called message passing. Then the neighboring node features are aggregated to create new node features. Repeating this process, node features are updated iteratively. My main GNN model is 3-layer graph isomorphism network. The intermediate representation of each layer are concatenated. And doing global readout, here I use summation and extracting the concatenated feature of the node where the mutation occurred and get the graph level representations. Then the graph level representations are input to multi-layer perceptron to get prediction. This is the overview of my approach. Then I assembled the prediction from several public notebooks and my GNN model. Before ensemble, the predictions of each model were converted into ranking. And here is one thing to note about the first place submission. The GNN model I use for first place submission contains bugs. First, the GNN model output almost identical predictions for all samples in each mini batch. That is to say, if batch size is 16, the prediction value changes only every 16 samples. Second, instead of wild-type features and mutant features being input to the model, wild-type features and wild-type features were input. That is, information on the type of mutation is not used. Luckily, these bugs contributed to the robustness of the predictions. In this section, I will share an interesting findings from this competition. I found there is some correlation between the centrality of the node where the mutation occurs and DTM. Centrality is a measure of the importance of each node. This image is a graph with colored by information centrality. This plot shows the relationship between information centrality and DTM in some training data. The inverse of information centrality scored 0.37 in public readable and 0.425 in private readable. I think that centrality may be related to surface area and residue depths. I will share my simple model score. My best single GNN model scored 0.365 in public readable and 0.368 in private readable. And treating the degree of each node as a categorical variable and using its embedding vector as a node feature, instead of embedding vector by ESM2, GNN model scored 0.192 in public readable and 0.389 in private readable. From this result, I think that information about the 3D structure of the protein converted into graph can be useful and GNN can extract it. Okay, that's all of my solution. My winning model is largely due to luck, and so there may not be much insight from my solution, but I hope you find it interesting to treat proteins as graphs. I enjoyed this competition very much. Thanks to the hosting team of this competition, and thanks to Kaggle team for this for the smooth organization of this competition. Thank you for listening.
Thank you very much. That was very impressive, and congratulations on the first place. Can you share a bit your graph neural network model must have made a difference? So you did an example of 5 different thermonet. There's some AlphaFold contributions. If you didn't have your GNN model in, would you have won the tournament or the challenge? or did it make the difference? Yes.
Sorry.
You know, if your graph neural network model was not included, would you have one?
Okay, and some the score of is that graph neural network. No, wait. Sorry.
Thank you, Maggie.
sorry.
Does that catch your question?
Yes, thank you. I will also write. Yeah.
Sorry. Please wait. I will chat comments. Sorry. Oh, okay. So how important was GNN to my solution? Okay. So my so wait a minute. I check my score. So well, my ensemble without my GNN scored private. Called in private 0.524. So this score is in leaderboard where about 20s in leaderboard. So if without GNN, I can't win this competition. And in my ensemble, and I checked, I removed each single model in my ensemble and compared the score. Rosetta model contributes most. And next is my buggy graph neural network model. So my graph neural network model is contributed some to score.
So it made all the difference. So
Yes.
that is good to hear. Can I ask, maybe I should write, but can I ask your background besides this protein dataset? Did you work with proteins before?
No, I know little about biology and protein. So I have little knowledge about protein.
Wow, it's very impressive. in 3 months to learn so much.
So yeah.
Guys, do you have questions? No?
I think we're good.
Can I ask the strategy for selecting the final submission? How did you have any strategy, or did you just take the best public performer ensemble, or how did you choose?
Oh, my first place submission is my best model in public readable. So I choose it.
Okay, thank you. And how did you decide on the weights, the individual weights of the individual single models?
Oh, I tried sometimes some weights combination, but and my first place submission, the weights are the best. So I don't assume anything about answerable weight.
Okay. Besides the methods you used in the final model, did you try many different models, many different other additional single models or
Sorry,
methods? I can write.
So
If I could just present a quick screen. So these are the submissions, and it looks like he labeled some of the different attempts. Is that right? You labeled your submissions with the different
yes.
Attempts that you made, over 200 submissions across the competition.
So you tried a lot.
It's bad.
Wow.
But I spent many of time to fix my bugs. So many of my submissions contains bugs.
Mm.
It was a good bug in the end. It got the first place. It can't be bad.
Many discoveries are realized by mistake.
Okay, any other questions, guys? Nope.
And you have his email. If there are any other questions, you can send him an email as well.
Yes.
Yes.
But thanks a lot and congratulations. It was very impressive to follow during the 3 months, and it's the effort you put in. It's it's really impressive.
Yes, thank you so much.
Yeah.
Thank you for your participation.
Thank you and congratulations.
Congratulations.
Thank you, dear.